Good afternoon, everyone. Thank you for the invite. It's great pleasure to share our research work with you all. I'm here to discuss about my project, Kynurenine pathway in multiple sclerosis. Before moving into the project, I would like to explain how the tryptophan metabolism happens in the central nervous system. In the periphery, tryptophan is circulated via blood in the albumin-bound form. Since tryptophan has higher affinity for the blood-brain barrier transporter LTAA than albumin, almost 75% of albumin-bound tryptophan likely dissociate from albumin to be taken up into the brain. Within the brain, most of the cells use it for protein metabolism. Neurons digest the tryptophan to serotonin and tryptophan, whereas 95% of the tryptophan converted via kynurenine pathway. This is the simplified version of kynurenine pathway for tryptophan catabolism. Tryptophan is one of the 10 essential amino acids responsible for production of NAD for cellular activity. The oxidative metabolism of tryptophan leads to an increased level of L-kynurenine by activating IDO or TDO enzymes. The cardinal role of kynurenine is to serve as the precursor of the neuroprotective kynurenic acid and picolinic acid, and the neurotoxic compounds 3-hydroxykynurenine and quinolinic acid. So, why these individual metabolites are important in central nervous system during disease state? This study demonstrates that changes in the brain levels of certain Kp metabolites may be important mediators contributing to brain cell dysfunction in some neurological disorder involving increased Kp activation. Black bar is neurons and white is astrocytes. The left side graph represent NAD plus production and the right side one is LDH release. We hypothesize that while some of the extracellular Kp metabolites may be beneficial for intracellular NAD plus synthesis and cell survival at physiological concentration, but they may contribute to neuronal and astroglial dysfunction and cell death at pathophysiological concentration. In this study, we found that treatment of human primary neurons and astrocytes with 3-hydroxyanthranilic acid, that's 3-HAA, and 3-hydroxykynurenine, that is 3-HK, quinolinic acid, quin, and picolinic acid, pic, at concentrations below 100 nanomolar significantly increased the intracellular NAD plus levels compared to non-treated cells. However, a dose-dependent decrease in intracellular NAD plus levels and increased LDH release activity was observed in human astrocytes and neurons treated with 3-HAA, 3-HK, quinolinic acid and picolinic acid at concentrations more than 100 nanomolar, and kynurenine at concentration above 1 micromolar. This study shows that not only these metabolites are important for producing cellular energy, it also shows that at pathological conditions, increased activation contribute to accumulation of toxins and cause damage to the cells. Even though kynurenic acid was discovered roughly 170 years ago, it was not until the 1980s and 1990s that the kynurenine pathway sparked substantial interest among neuroscientists. This was due to the discovery that the two major products of the pathway, kynurenic acid and quinolinic acid, possess significant yet opposing effects on various neuronal cells and physiological processes. The kynurenine pathway has been studied extensively since 1990, with a surge in the number of publications from 2010 to today, which becomes 3 times more than 2010, and it keeps increasing. Our group has characterized the kynurenine pathway in several brain cells, including astrocytes, microglia, and macrophages, and also neurons and oligodendrocytes. The Kp branches into two major pathways. one implicated in neuroprotection and the other in neurotoxicity, that are segregated across cell types. Under physiological conditions, the neuroprotective branch is more active as most of the kynurenine in the brain is metabolized into kynurenic acid, NMDA, and alpha-7 nicotinic acetylcholine receptor antagonist through the action of kynurenine aminotransferases expressed mainly in astrocytes, whereas under inflammatory conditions the metabolism is shifted through kynurenine monooxygenase to produce 3-hydroxykynurenine and other toxic metabolites such as quinolinic acid. This is an NMDA receptor agonist and oxidative stressor. KMO is primarily expressed in microglia and the resident immune cells in the brain, and is also expressed in high levels in peripheral immune cells such as monocytes and macrophages. The KP in oligodendrocytes demonstrated that IDO1 and tryptophan 2,3 deoxygenase are not expressed in human primary oligodendrocytes. This potentially has strong implications for MS pathology. The lack of these two KP regulatory enzymes in oligodendrocytes is associated with higher cellular susceptibility to allogenic T cell challenge, since IDO1 plays a crucial role in immune regulation, particularly in suppressing T cell proliferation. Before moving to main part of the study, I will provide brief introduction about MS. Multiple sclerosis is a chronic disease of central nervous system with persistent inflammation and demyelination resulting in varying degrees of neurological deficit. This is the most common disabling neurological disease affecting young adults and females. Approximately 2.8 million people worldwide are affected by this disease. The exact factors that initiate inflammation are still unknown, but it is generally believed that MS is caused by environmental factors in a genetically susceptible host that trigger a T cell autoimmune response against the central nervous system. Although viruses may trigger MS relapses, there is no definitive evidence that there is an MS virus or ongoing chronic infection of the central nervous system. Epidemiological evidence suggests that Epstein-Barr virus, an ubiquitous virus, plays a key role in MS. Oligodendrocytes are the primary neural cells affected in MS. So, we characterize the KP in primary oligodendrocytes and the cell line. We found that both primary oligodendrocytes and MO3.13 cell line did not express IDO. However, MO3.13 cells expressed TDO and thus is potentially able to catabolize tryptophan. Primary oligodendrocytes which lack IDO and TDO are unlikely to be able to degrade tryptophan. In order to test the quinolinic acid toxicity in vitro, we chose mouse oligodendroglial cell lines N19 and N20.1. Using immunohistochemistry, we have demonstrated the capability of both the cell lines to take up exogenous quine, and this indicates the quine is catabolized intracellularly in a time-dependent manner as fluorescence intensity was directly proportional to the uptake of quinolinic acid. We then further assessed the cytotoxic threshold of such quine on quine uptake on oligodendroglial cells. A standard curve using commercial exogenous quine to assess the level of gliotoxicity by quine on N19 and N20.1 cells. This demonstrated a relatively similar level of tolerance in the two cell lines to quine. There was a notable trend indicating dose-dependent quine toxicity on oligodendrocytes where the LD50 for N19 is 0.5 micromolar and for N20.1 is 1 micromolar for 72 hours. These LD50 values were chosen as a reference criteria for subsequent quine antagonism studies. To neutralize the quine toxicity, we have used the quine monoclonal antibody in this cell model. I have showed that quine monoclonal antibody neutralizing the toxic effect of quinolinic acid. Neurotoxicity of quine on oligodendroglial cell lines were decreased by increasing concentrations of quine monoclonal antibody. To analyze the KP, we have used the chronic progressive EAE mouse model induced with myelin oligodendrocyte glycoprotein and pertussis toxin. Clinical severity score was based on the approved clinical score. Score of 0 indicates normal. Score of 1 indicates limb, tail and or hind limb weakness. Score of 2 indicates both limb, tail and hind limb weakness. Score of 3 indicates partial hind limb paralysis. Score of 4 indicates complete hind limb paralysis and forelimb weakness. And score of 5 indicates moribund state. And this graph showing the clinical score of EAE mice and wild type control, and the disease severity also represented as area under curve. Compared to wild type control, there is a significant difference in the disease severity. We have also assessed the EAE associated CNS pathology in the spinal cord from mice at the peak of the disease and stained with HNE to assess the inflammation, and LFB and NISL to assess the myelination. Independent histological analysis of spinal cord revealed that there is an inflammation and demyelination in EAE-induced mice. To analyze the kynurenine pathway metabolism in the EAE disease progression, we have grouped these mice according to the clinical score. Cohort 1 indicates EAE score of 0.5 to 1, cohort 2 is 1.5 to 2, cohort 3 is 3 to 4. To find out whether the KP is activated in EAE disease mice progression, we have measured the kynurenine and tryptophan ratio as a measure of IDO activity. In CNS, this IDO activity is directly proportional to the disease severity. We have also observed that there was a significant difference between the least severe group compared to the most severe group, that is cohort 3 in both brain and spinal cord. This suggests that the induction of kynurenine pathway may be related to the progression of EAE. This trend is also observed in serum samples. The KP activation is further confirmed in IDO1 gene expression studies by real-time PCR in EAE-induced mice, brain and spinal cord. In injured site, the IDO is highly activated and the staining was more prominent. Based on this immunohistochemistry, we have confirmed that IDO is activated during inflammation in EAE-induced mice. We then measured the first diverging branch of kynurenine pathway. In the midstream branching, kynurenine aminotransferases 1 and 2 synthesize kynurenic acid from kynurenine pathway cardinal compound kynurenine. We have also measured the kynurenine monooxygenase to analyze the downstream catabolism. In both CNS and plasma of EAE mice cohort, there is a significant decrease in the production of kynurenic acid in comparison to the control. Within the CNS, the kynurenic acid is lowest in the spinal cord tissue of the most severe group compared to the early cohort, suggesting that the production of kynurenic acid may actually be compromised during disease progression. We then analyze the expression of two enzymes, namely CAT1 and CAT2, involved in the catabolic reaction of kynurenine to kynurenic acid by real-time PCR. Our results indicate that the expression of CAT1 and 2 were significantly downregulated, particularly in spinal cord tissues, in agreement with the observation of kynurenic acid content found in spinal cord tissue. Whereas in brain, only CAT2 expression was significantly downregulated. Further, KMO expression is more prominent in the spinal cord of EAE-induced mice. The last branching point within the KP occurs at 2-amino-3-carboxymucrate semialdehyde, where it gets either converted enzymatically to picolinic acid by enzyme ACMST or non-enzymatically to quinolinic acid. Again, quinolinic acid is metabolized to produce NAD by QPRT enzyme. Picolinic acid was significantly decreased in both periphery and nervous system of cohort 3 in EAE-induced mice group than in control. We also noted a progressive trend of subnormal concentration of picolinic acid from cohort 1 to 3, suggesting a correlation between decreased production of picolinic acid and EAE disease progression. Quin, on the other hand, was significantly increased in both periphery and central nervous system of EA-induced mice in comparison to the control. This effect was more prominent in the spinal cord tissue with trend of increasing queen production as the disease severity increases, that is from cohort 1 to 3. Furthermore, the expression of ACMSD was significantly downregulated in comparison to the control, thus validating this decrease of picolinic acid production in both brain and spinal cord tissue. Surprisingly, we found QPRT was significantly upregulated in CNS, indicating increased activity in the catabolism of quinolinic acid in EA-induced mice. This was further validated by immunohistochemistry of QPRT. In our study, queen concentration remains high in both periphery and central nervous system, suggesting that QPRT is very likely to be saturated, resulting in the accumulation of quinolinic acid. Furthermore, the levels of queen did not reach pathophysiological toxic threshold as of those found in other neurological diseases, suggesting that queen is chronically accumulated in the progression of EAE and may involve in neurodegeneration. We have also measured the NAD+, and NADH ratio and plasma NADH in healthy and EA-induced mice. We found that NAD+ and NADH ratio were significantly decreased in EA-induced mice. This shows that QPRT is saturated due to increased accumulation of queen and not able to produce enough NAD+ for the cellular energy. In order to test which cell type is responsible for the overproduction of queen in EA-induced mice, we have done the double staining with neuronal markers. We have found queen is co-expressed with microglial and macrophage cells, but not with neurons, astrocytes, or oligodendrocytes. This shows that monocytic cells of the innate immunity, when activated, can produce pathophysiological amount of quinolinic acid suffice to induce neuronal cell death. It is also plausible that queen from these monocytic cells are secreted exogenously, which is then taken up by surrounding neural cells for catabolism, eventually saturating the QPRT level within those cells, contributing to accumulation of queen that subsequently leads to neurotoxicity. This may also partially explain for the observation of gradual rise in queen to reach its pathophysiological concentration given the short duration of disease progression. This is highly relevant in the context of chronic neurological diseases, particularly since damage from MS during early stages is mainly due to immunological dysfunction inflicted by adaptive immunity, while later stages of disease progression exhibits a distinct classic neurodegeneration manifestation corresponding to the activity of innate immunity. We have also assessed the kynurenine pathway in multiple sclerosis progression in human plasma samples, and we have found a similar trend in those MS patients. Several drugs that modulate the kynurenine pathway are under investigation by our laboratory and others. For example, for depression, for chlorokynurenine, and for myalgic encephalomyelitis, chronic fatigue syndrome, kynurenine, and there are several IDO inhibitors for phase 1, 2, 3 clinical trial, cancer clinical trials, and chemo inhibitors has been used successfully for preclinical studies. Kynurenic acid analogs are about to enter clinical trials for the treatment of epilepsy, a stroke, and possibly Parkinson's disease. And 2 KP analogs are in MS treatment, teriflunomide and lakinamide. And KP analog has reached the market in Japan as a potent immunomodulative drug for the treatment of MS, asthma, and dermatitis. In order to test KP modulators effect in EA-induced mice, we treated the EA-induced mice with 1-methyltryptophan IDO1 inhibitor, 100 mg per kilogram body weight for 7 days intraperitoneally after the mice reached a score of 2. We found that 1MT treatment significantly decreased the clinical symptoms within 3 days of treatment, followed by the transient reduction disease symptoms during day 6 and 7. Next, we modulate this pathway at mid-level by blocking the KMO using RO6218048 at the dose rate of 100 mg per kilogram body weight for 7 days intraperitoneally. Our 61-80-48 treatment significantly decreased the disease severity compared to control. In order to test whether kynurenine has effect on multiple sclerosis, we treated the mice with kynurenine 200 mg per kilogram body weight intraperitoneally every day until day 10 at the same time as EAE induction. Red double arrow indicates the treatment period and the black arrow indicates the animal monitoring using standard clinical scale. In the left side of the graph, we showed that the mean clinical score of vehicle control, kynurenine treatment, and the blue indicates the AHR ligand positive control TCDD. The right side mean clinical score AUC showed that there is significant difference in mean clinical score between vehicle and kynurenine treated mice, and there was no significant difference between the kynurenine and TCDD. The prophylactic kynurenine treatment significantly decreased the disease severity and subsequent relapse. In a more clinically relevant treatment paradigm, that is therapeutic kynurenine treatment, otherwise called initiating the treatment after the clinical score of 2 and above, we have shown that there is not significant difference between vehicle and kynurenine treatment until 36 days. But after 36 days, kynurenine treatment prevented the relapse in EA-induced mice. The present study demonstrates for the first time that there is a parallel link between the degrees of chronic EAE clinical deficit and the levels of KP metabolites. This study further supports our data from the human MS samples and demonstrates that the KP metabolism is involved in the progression of MS patient and EAE mouse model. Increase in IDO activation and change in the production of bystander catabolites from the KP through the course of EAE disease progression show that this pathway is activated differently at different stages of disease severity. Appropriately timed KP modulation is as part of the MS treatment helps to slow and possibly halt the disease progression. So these are our ongoing studies looking at a kynurenine pathway gene variants in MS patients and conditional knockout mouse models for KMO and QPRT. And there is a queen monoclonal antibody treatment in 3D organized cell culture with our collaborators. And I would like to thank Professor Bruce Prue and Professor Jill Chiliman. And I would like to thank my lab colleagues and my collaborators, my funding bodies. If you have any questions, please contact me through my email ID. I will answer as soon as I can. Thank you very much.